Okay, now we really are on live streaming on Facebook, and I guess I will share my screen. And maybe I start. Dear all, welcome to this two-day meeting on Down syndrome from research to therapy. And first of all, a warm welcome to our dear friends who are connected from different countries. And a warm welcome to all Italian researchers. I see that there are very many. And welcome also to non-scientists, families, and relatives of people with Down syndrome who will be able to attend this meeting either now on Zoom or via Facebook with the live broadcast that Gianfranco Salvini has helped me to organize for us. Now, this meeting, as you all know, is organized by the Down Syndrome Task Force. And the Down Syndrome Task Force is a group of scientists, of 12 scientists. You see they are listed here, plus the president of the two most important Italian Down syndrome associations, AIPD and CoreDown. What is the purpose of Down syndrome? Other than organizing this meeting, the purpose of Down syndrome is to increase the quantity and the quality of scientific research on Down syndrome, promote scientific dissemination and interaction between researchers, associations, and families, and promote and participate to activities that can improve the quality of life of people with Down syndrome. Dissemination has been one of the most important aims, and it has mostly been done by organizing online meetings that we call Scienza incontra Down, Science meets Down syndrome people. Over the years, we have been treating several subjects that were discussed by a number of scientists that agreed to participate. Now, let me say that the day of the meeting today is a sort of celebration for us. We managed to organize this 7th National Italian Scientific Meeting, a celebration because the meeting is our most relevant activity. And our meeting today will start with an international session. This is a very important session for us because it is our way of focusing on the relevance of international cooperation. And this is a very important issue, even more important these days when we are losing the good of intellect, as our big poet Dante Alighieri wrote in the Divina Commedia, and the stupidity of war rages around the world. I personally think that it is important, both as a scientist and as a man, not to miss any opportunity to fight for peace, to say even one simple word in favor of peace. I'm very glad to see today colleagues from different countries. Hope this international session will be successful and hope that will encourage us to continue on this path. And now I leave the microphone to the two persons who strongly wanted and organized this session. Thank you, Renata. Thank you, Marzia. Please open this international session.
Thank you, Lucio. I'm wondering whether I can share my screen just to introduce the session, if possible.
Yes, you can.
Can I help me in case I share screen? Okay, screen. Okay, can you see? I hope that you can see my screen.
Yes.
Right. Okay.
You can put it in presentation if
yes.
you...
Where is the knob? Presentation.
Down to the right. Over there, close. You were very close. Down to the right.
Down to the right.
At the bottom.
Okay, here I see it. Okay, okay. Ah, no, normal. Okay. Okay, right?
Good.
Okay. Good afternoon. Marcia Perluigi and I are very pleased and honored to introduce and moderate this symposium, international symposium, entitled, as you can see, International Efforts to Understand Down Syndrome Pathology and Seek Therapeutic Interventions. This symposium covers a wide spectrum of topics, all of utmost importance in the field of preclinical and clinical research on Down syndrome. Thus, we do really hope that you will enjoy this session. The speakers of the first part of the session are well-known Italian researchers who have obtained important achievements in various fields of relevance for Down syndrome, going from genes, aging, and behavior. The talks will last 30 minutes each, 20 minutes for presentation of the data, and 10 minutes for discussion that will take place at the end of each talk, altogether 30 minutes. You can directly ask questions after each talk by raising your hand through the hand tool of Zoom. In my browser, it is at the bottom of the screen on the right. I don't know, it depends on the browsers. Okay. Take place at the end of each talk, altogether 30 minutes. Or alternatively, you can write your questions using the chat tool of Zoom. And now I think we can start with the first session. The first speaker is Simona Palladino from the University of Naples, Federico II. Her talk is entitled A Dangerous Liaison Among Endosomal Pathways, SYNJ1 and Down Syndrome. Please, Simona, the floor is yours.
Stop sharing.
Yes, I stop sharing, yes. Done. Okay.
Okay.
Simona, please.
Thank you very much. I'm trying to share my screen. Do you see my screen?
Yes.
Okay,
Perfectly.
perfect. Okay. So I just I don't remember this to remove the annotation that Professor Nietzsche said to me yesterday, but I didn't.
Don't worry about it. Go ahead.
Okay.
Don't worry.
Okay.
If something happens, we'll take care. Go ahead.
Okay. Thank you. So, sorry. Good afternoon again, everyone. And first of all, I would like to thank the organizer for giving me the opportunity to present our data. And today, I wanted to tell you the story of a dangerous liaison we have recently unraveled among endosomal trafficking, SYNJ1 and Down syndrome. Indeed, the outline of my presentation will run through these three key items. I don't need to say anything about Down syndrome. We are in an audience of experts, but just a few considerations. The complex multi-organ disorder of Down syndrome mirrors at the cellular level the perturbation of multiple pathways so that they may concur to disease pathogenesis. And on the other hand, each pathway may be involved, more than one pathway may be involved in one clinical phenotype. Sorry, because I have a problem. Okay, sorry, because I don't see my screen for Intel. Okay, it's okay. So, moreover, lots of studies have shown that the dysregulation of several genes may concur to Down syndrome pathogenesis. So keeping in mind this consideration, we move to the second item. Endolysosomal trafficking is essential for cellular homeostasis and as well for cellular plasticity. That is a fundamental property of each cell type to meet physiological needs and to dynamically address different stimuli. In fact, the endolysosomal system is a critical cellular hub that regulates the exchanges with the cellular exterior by controlling uptake of nutrients, cellular communication. It modulates cell signaling. It guarantees proper composition of the cellular compartment. And finally, endolysosomal system controls the molecular turnover and the cellular proteostasis. Compelling evidence has shown the neurons seem to be more sensitive to endosomal trafficking perturbation. And on the other side, the dysfunction of such pathways have been associated to several neurodegenerative diseases, altogether supporting a critical role of these housekeeping pathways for neuronal physiology. So the third item, SING1 gene. SING1 is one of the 100 genes located on chromosome 21. It encodes for the inositol phosphatase synaptogenin 1, a protein a long time ago in 1994 discovered by the Camillis group as a protein enriched in the presynaptical terminal of mice. Interestingly, SING1 has a much wider distribution in humans. Among inositol phosphatases, SING1 is peculiar because it has two catalytic domains. The canonical SAC1 domain common to all phosphatases, and the central region, the so-called 5-phosphatase domain. Thanks to this double catalytic activity, SING1 can act on different substrate as depicted here, and as a consequence of these unique characteristics, SING1 can be implicated in a plethora of cellular process. And on the other side, the dysregulation of SING1 may affect different cellular pathways. Like whole inositol phosphatase, SING1 acts in concert with the specific kinase together to control the proper level of this class of lipids. For their selective organelle distribution, phosphoinositides have emerged as key regulators of membrane trafficking. Indeed, they can organize and hold the organelle identity by selectively recruiting proteins and or effectors to specific organelle, as well by interacting with the membrane proteins. The kinases and phosphatase themselves can take part to membrane trafficking regulation by controlling the spatiotemporal compartmentalization of these lipids. And we have recently highlighted that SING1 is a key regulator of endosomal trafficking. Specifically, SING1 regulates the homeostasis and function of early endosomal compartment in different cell types. Indeed, the loss of SING1 activity as a consequence of the knockdown of the SING1 expression or of the expression of the pathological Parkinson associated mutant, leads to the enlargement of early endosome and the impairment of the recycling pathway. So on the basis of these premises, but also taking into account the previous observation showing in somehow alteration of the endosomal compartment in cells from adult individuals with Down syndrome, we decide to explore if whether there is an interplay between endosomal trafficking and SING1 in Down syndrome, and if yes, which is and when. So to make a long story short, through a deep imaging analysis combined with the biochemical approach, we figure out this functional link. Specifically, we have shown that the structure and the dynamics of early endosome are altered in human trisomic fetal fibroblasts. I just show you one of the key experiments. Early endosome labeled by the marker RAB5 are occupy a larger area of the cell in trisomic cell, as you can appreciate in 3D reconstruction. They are larger in terms of size, and they are brighter in terms of fluorescence intensity, as you can appreciate in the intensity map in the graph. Moreover, we have demonstrating that the recycling pathway is markedly compromised in trisomic cells, while no alteration in the trafficking toward the lysosome. I want to stress that these alterations are present already at the fetal stage, thus indicating that they occur very early during development. And altogether, these data support that the dysfunction of endolysosomal pathway may concur to the pathogenetic mechanism of Down syndrome. So next question is, which phenotype of Down syndrome could be affected by endosomal dysfunction? And as I present you on the basis of what I said to you before, we decided to investigate whether endosomal dysregulation may account for defects in neuronal development. So to this aim, in collaboration with Antonella Rizzo and Lucio Nitsch, we took the advantage of novel and efficient experimental procedures to differentiate induced pluripotent stem cells. And specifically by using a dual SMAD inhibition protocol, isogenic euploid and trisomic iPS cells were differentiated to neural progenitor MPC at the very early stage 7 days and at the late stage 21 days. And so by applying a combined approach, imaging and biochemical approach, we have analyzed whether and when the alteration of endolysosomal pathway occur during neural differentiation. So first, we found that the homeostasis of early endosome is altered in trisomic iPS cells. Here I show you the early endosome labeled with another marker of this compartment, EA1, by using a specific antibody. And as you can see in the single confocal section, and well, you can appreciate in the 3D reconstruction, EA1 positive structure are much brighter in the trisomic iPS cells and also larger, thus indicating an enlargement, an expansion of this compartment. Interestingly, by Western blotting analysis, we found that the levels of EA1 are higher in trisomic IPS than euploid ones, thus suggesting an alteration of the dynamic of this protein. We obtained the comparable data by looking at the key regulator of early endosome biogenesis, that is RAB7, by bioimaging approach as well by biochemical approach, further strengthened the alteration of this compartment, the dyshomeostasis of this compartment in trisomic IPS cells. On the contrary, we found no alteration in the structure of late endosome labeled using a specific antibody labeling RAB7, a specific marker, sorry, for this compartment. And consistently, we found comparable levels of RAB7 by immunoblot analysis in euploid and trisomic cells. Thus, altogether, these data clearly indicate a dyshomeostasis specifically of early endosomal compartment at undifferentiated state. And so what happens during a neural differentiation? At the early stage, as you can see in this picture, RAB5 positive structure result much brighter and further indicating, supporting an expansion of this compartment. And consistently, we found a higher level of EA1 and RAB5 by immunoblot analysis. That's supporting that early endosome are still altered at this stage. Interestingly, at late stage of differentiation, we found a drastic alteration of EA1 and the RAB5 positive structure, as you can see in the single confocal section and much well in the 3D reconstruction. That's still indicating a dyshomeostasis of the compartment. But then I want also to show you that in trisomic fibroblasts display at least two populations of endosome in terms of early endosome, in terms of fluorescence intensity. So that's suggesting an alteration of the dynamics of this compartment that is supported again by the high level we found by immunoblot analysis for EA1 and RAB5. So at this point, the question is whether these early endosomes are functional. And to address this question, we analyze the recycling pathway by monitoring the trafficking of transferrin receptor. And by using a cold pulse and chase assay, I briefly explain here. Cells were incubated with the fluorescent transferrin for 7 minutes. That is the pulse. Then transferrin was washed out, and cells were chased for different times using a normal culture medium. As expected, in euploid progenitors, transferrin fluorescent signal decrease over time, as you can see, almost nothing at 20 minutes of chase, because the transferrin together its receptor recycling back to the plasma membrane. On the contrary, in trisomic progenitors, the fluorescent signal of transferrin remains still higher at 20 at the same chase time point. You see there is an accumulation of transferrin, is also clear in the quantitation, so that's indicating an impairment of recycling pathway. Thus, altogether, these data clearly indicate that homeostasis and function of early endosome are impaired along neural differentiation. But at this point, the question is, which is the role of SING1 in this story? And the SING1, we found that is upregulated in trisomic cells at the fetal stage and also at an undifferentiated state, supporting its involvement. But the key experiment proving its role is the experiment in which we overexpress SING1 in the unique available human neuronal cell model, this very strange name, SHSY5 cells. So the SING1 overexpression in these cells recapitulates the alteration observed in trisomic cells, directly proving its role. And so indeed, we found that SING1 overexpression affects homeostasis of the early endosome and also affects the recycling trafficking. Altogether, these data clearly indicate the detrimental link between SING1 and endosomal pathway in Down syndrome. But also, these data highlight the crucial role, the critical role of SING1 for neuronal cells. And to support this role, interestingly, we found an increase of SING1 expression during the neural differentiation. So at this point, the key question is, how does SING1 dysregulation and in turn endosomal alteration lead to neuronal perturbation? To address this question, in collaboration with Giuseppe Merla in our department, we decided to perform RNA-seq analysis comparing wild type and SING1 overexpressing neuronal cells. And interestingly, we have found several genes that are deregulating upon SING1 expression, both up and down. And among them, very interesting, we found genes that are that control the synaptic transport, regulate level and transport of neurotransmitted, but also genes implicated in neurogenesis, in axonal guidance. So altogether, supporting the critical role of SING1 for neuronal physiology and likely by modulating neuronal plasticity. Finally, on the clinical perspective, we can say that our data suggests that endosomal pathway may be a new a novel therapeutic target for Down syndrome by attenuating SING1 expression and/or modulating its activity. To support this hypothesis, we found that the knockdown of SING1 expression in trisomic fibroblasts restores the homeostasis of early endosome. So in conclusion, our data overall indicate that early dysfunction of endosomal pathway may concur to Down syndrome neuropathogenesis, And they also point out a critical role of SING1 in DS pathogenesis. And let me conclude saying that the proper levels of SING1 are critical for neuronal homeostasis. So I hope to have convinced you of this dangerous liaison and putting in this way a new piece of the DS puzzle. Obviously, there are other pieces that altogether we have to understand. And so the last but not the least, I want to thank the people that work with me at this project, and a special thank to Laura De Rosa and Dominga Fasano, who take care of this project, Andrea Conte for the RNA-seq analysis, the long-lasting fruitful collaboration with the Giovanna Pierantoni group, and also the long-lasting collaboration, very strict collaboration in the last period with the Lucio Nicci group, Antonella Izzo, Nunzia Mollo e Anna Conti, all the other, Adriana Limone e Daniela Sarnataro for helping in neural differentiation protocol, all the other collaboration, and obviously you for the attention. Thank you.
Thank you very much for your beautiful talk. And the discussion is open. Let me see whether there are questions in the chat. Yes. Yes. Very interesting. Have you checked late endosomal compartment in fully differentiated iPS cells? Is it possible that over time, the early endosomal pathology reverberates to the late endosomal, lysosomal, exosomal compartment as suggested in murine models of Down syndrome?
Okay. Thank you for this question. Yes, we have already analyzed what happened to the late endosome during neural differentiation. And the preliminary result, I said preliminary because we got this interesting data, but we want still to go inside. The data is that seems that different on what happened in iPS cells, where late endosome are completely unaffected. In the trisomic cells in the progenitor at 21 days, there is like a depaupation of late endosome. So also by immunoblotting analysis, we found low level of RAB7. So this is very interesting because is telling another important issue of probably in neuronal cell during the neuronal differentiation, that when we have this block at the level of the early endosome, because we know that there is a very important dynamics of maturation between early to late endosome, this is probably not occurring in neuronal cells. This is why I say I wanted to underline that is very interesting, because this is doesn't happen in fibroblasts. So probably there are a regulation that is a specific regulation for neuronal cells. And this is also what in the near future we would like to understand. And just for exosome part, this is also a very interesting point. This is what we are starting doing to see, at least we have a preliminary experiment comparing the medium of different euploid and trisomic cells. It just because in the question there is also about the lysosome. For the lysosome, I didn't have the time to show you, but we have a very I just can show you now. We have just a very key experiment showing that lysosomes are altered in trisomic, in this case, fibroblasts in terms of area occupied, in terms of the size, as you can see also in the ultrastructure. These are all LAMP1 positive, so lysosomal protein labeled. And also it's very interesting that there is a displacement of lysosome, you see in euploid, with respect to trisomic cells. And this displacement is due to the collapse of early endosome to the center of the cell.
Okay, there is another question. The question was put, the former question, by Pasquale da Corso. Now there is a question by Jacqueline London. Is synaptogen SINJ1 expressed in other cells than neuronal, and how in Down syndrome?
Yes, is expressed. This is important. I stressed at the beginning, probably not much. This is a difference between the mouse and the humans, because in the mice there is a very high enrichment of SINJ1 in the brain, in different areas of the brain, while in the humans there is a much wider distribution. Also, for example, very interesting, I mean, for my previous story in the research, also in thyroid gland, but is expressed ubiquitous. And we don't know if there is a different regulation of the in terms at different level, transcriptional and at the protein level. Even if we know that SINJ1 undergo to phosphorylation, and in particular, dephosphorylation is important for its activity. So, and in trisomic, the data that I show as a scheme for were performed in trisomic fibroblast, fetal fibroblast, human fetal fibroblast. Okay, so this is the alteration is in human fibroblast.
I don't see any other question in the chat. I don't see hands, also possibly because you have not unshared your screen. We have still 30 seconds. Okay, I don't see raised hands. No, I have a very short question, and naive possibly, if I can.
Thank you.
How does synaptogenin modulate the expression of such a large number of genes? The mechanism, any idea of how? Because it seems very...
Yeah.
Okay.
Thank you, Serenata. No, thank you, Serenata, for this question. The quick answer is we don't know perfectly, and so we wanted to understand this. But my idea is that our idea is that when we impair this important pathway, cellular pathway in neurons that are terminally differentiated, probably these start to change the transcriptional program of the cell. So this is what we wanted to try to understand. And probably there are also other regulation also at the epigenetic level. But for sure, SINJ1 controlling these different phosphinosytides that controls the endocytosis by 5-phosphatase domain, the endosomal trafficking by SAC1 domain, and also we have in collaboration with the Paolo Remondelli demonstrated in another model system in the Parkinson fibroblasts from a PARC20 patient, that there is also a new alteration of the early secretory pathway. So it's possible that these different the alteration of these different cellular pathways then in turn in neuronal cells become completely detrimental.
Okay, thank you very much. If there are, Simona, for a very interesting talk, and
Thanks.
if there are no other questions, I think we can move on to the second speaker of this afternoon, Maria Giulia Baccalini from IRCS and Institute of Neurological Sciences in Bologna, Italy, and her talk is entitled Trajectories of Atypical Aging in Down Syndrome, Insights from Epigenetics. Maria Giulia, please.
Okay, good afternoon, everybody. Can you see my screen?
Yes, thank you.
Perfect. So, thank you, and I would like to thank the organizers for giving me the opportunity to present you this data that we are starting to generate on the relationship between epigenetic changes that occur in Down syndrome and the typical aging of people with Down syndrome. So, our story starts several years ago in 1975 when George Martin published this pioneering work in which he proposed that Down syndrome is a progeroid condition because it has several phenotypical traits characteristic of aging. And today we know that people with Down syndrome actually experience an atypical aging as they manifest with a higher prevalence some age-related traits, in particular at the level of the immune system with a profile which resembles the one of immunosenescence, and at the nervous system with the well-established higher prevalence of Alzheimer's disease. Now, if we want to study aging in people with Down syndrome, we should refer to those that the researchers in the aging field call the hallmarks of aging or pillars of aging. This is a set of molecular mechanisms or cellular processes which have been demonstrated to be profoundly altered during aging in the general population, and whose modulation can affect the rate of aging, promoting or delaying it. And among these hallmarks of aging, the one in which we are interested in are the epigenetic alterations. And in particular, we are interested in DNA methylation, which is the most studied epigenetic modification. Basically consists in the addition and covalent addition of a methyl group to the cytosine of the DNA, usually a cytosine which is followed by a guanine. So we talk about CpG dinucleotide. And DNA methylation is very important in regulating the structure of the chromatin and therefore the gene expression. It's established early during development and plays an important role in tissue differentiation. But what is important for us is that epigenetic DNA methylation is largely remodeled during aging, and that a series of environmental factors can further modulate it. So, so far, many studies have investigated the DNA methylation of the genome, what is called genome-wide DNA methylation in Down syndrome. These studies have considered different tissues, so for example, blood, brain, cellular models, and have included participants of different ages, so newborns, children, adults. And all these studies have reported a widespread alteration of DNA methylation patterns in Down syndrome. However, only few of these studies have considered the relationship between the epigenetic alterations and the aging phenotype of Down syndrome. And in particular, they not considered how this DNA methylation alteration that we find in Down syndrome behave during aging. So the aim of the analysis that I will present you is to investigate how the epigenetic alterations that characterize Down syndrome are remodeled, behave during aging, so their aging trajectories. And to do this, we generated a new data set of DNA methylation data on a cohort including 54 participants with Down syndrome and 42 controls. All the participants were enrolled in 2 centers, in Troina and in Bologna. And the participant with Down syndrome underwent an extensive clinical and neuropsychological evaluation. What is important of our cohort is the age range of the participants, because as you can see, it's quite wide, goes from more or less 20 years to 60, 70 years. So having this wide age range, we are able to track the trajectories along aging. And the other nice thing of our cohort is that for a subset of samples, 17 controls and 24 people with Down syndrome, we have 2 longitudinal time points, which are on average 13 years apart. So this can allow us to track the longitudinal change in DNA methylation in the same individual. All these samples were analyzed through the Illumina microarray EPIC, which is a microarray which returns the DNA methylation value of more than 850,000 CpG sites widespread across the genome. And the data were analyzed through basically linear models, correcting for potential confounding effects like sex and blood cell counts. Okay, so we organized our analysis in 3 steps. In the first step, we wanted to compare, to understand the relationship between the DNA methylation change that characterize Down syndrome and those that occur during physiological aging. So I explain better. The first thing that we did is to perform 2 comparisons. On one side, we compared the DNA methylation in people with Down syndrome respect to euploid controls, and we identified a lot of differentially methylated position. And then we considered only the controls, and we looked for those CpG sites whose DNA methylation is associated with aging. So the sites that are associated with aging in the general population, in the controls. And also here we found a long list of differentially methylated position. And then we intersected these 2 lists in order to identify those probes that satisfy both the comparisons, so that have a DNA methylation level which is different between participants with Down syndrome and control, and a DNA methylation value which change with age in the general population. And we found these 10,000 more or less of probes of CpG sites. And the interesting thing is that this intersection, the extent of this intersection is higher than expected as a result from the Fisher-Exact test, which returned a very significant p-value with a high odds ratio. So in other words, the CpG sites that have a different methylation between people with Down syndrome and controls are enriched in CpG site in genomic location whose DNA methylation normally changes with aging. And then we look at the functions of the genes in which these CpG sites map, and we found that these 10,000 CpG sites map, and we found that there was an enrichment in biological processes related to development and morphogenesis, together with a regulation of the gene expression. So from this first set of analysis, we can say that the genome-wide alterations in DNA methylation that occur in Down syndrome are enriched in genomic regions that, first of all, have a DNA methylation profile that changes with aging in the general population, and that map in genes which are related to developmental process. How can we interpret these 2 results? We can interpret in part them looking at the theories of aging. So you know that during the history of research, several theories have been proposed to explain basically why do we age. And these theories can be divided in two groups. On one side, we have the damage-based theory, which are actually the most widely accepted today. They state that during aging, there is the progressive accumulation of damage at the level of nucleic acids, proteins, lipids, and so on. And that this damage leads, this progressive accumulation of damage leads to a progressive decline in the function of our body. And then we have a set of theories which are called programmatic theories that states that aging derives from somehow predetermined mechanisms. And in particular, some authors sustains that basically aging starts in development, and that during development there is a set of genetically regulated programs which are very important for development and for a successful development. The problem is that these developmental programs are not switched off after the end of development, let's say after the reproductive time, but they continue along the entire life of the individual, and at a certain time they become detrimental and cause aging. So in this view, we can hypothesize that from an epigenetic view, the atypical aging of Down syndrome is intrinsic in the development alteration caused by the trisomy of chromosome 21. So the trisomy of chromosome 21 somehow alters some of these genetic programs that are not important only for development, but that cause aging later in life. And in support also of this hypothesis, we look at how behave this set of 10,000 probes, CPG sites, in two data sets that were available in the literature regarding DNA methylation in newborns and children with Down syndrome. And you see that although we selected these CPG sites in a cohort of adults and to be associated with aging in adults, they are still able to basically divide perfectly people, the children with Down syndrome and the controls. And this means that they are already altered at the level of birth and possibly also before. Okay, so this first set of analysis tells us that there is a relationship between aging and Down syndrome. But the next question is, how do the DNA methylation chains associated to Down syndrome behave during the aging of people with Down syndrome? So what we have done is to add a comparison in our analysis, and basically we considered only the people with Down syndrome, and we search for those CPG sites that have a DNA methylation value which is associated with age in people with Down syndrome. And also here we found a lot of differentially methylated positions, but here for this presentation we are interested in this subset of CPG sites which are at the intersection between the three comparisons and therefore have a DNA methylation value which is different between people with Down syndrome and control, and the DNA methylation value which is associated with age in both controls and people with Down syndrome. We analyzed this set of CPG sites through different approaches, but what I want to show you is the behavior of this CPG site, which is representative of the large part of these 5,000 CPG sites. So you can see that this, you can see that this CPG site is hypermethylated in people with Down syndrome, the orange, respect to controls, and that it's hypermethylated with age in both controls and people with Down syndrome. But if you look at these two lines, which are the regression lines of DNA methylation with respect to age, they're basically perfectly parallel. So it means that the DNA methylation, the rate of changing of DNA methylation during age is the same in people with Down syndrome and controls. And this is evident both when we look at the cross-sectional sample And when we look at the longitudinal sample, in which each line is basically an individual and the two dots are time 0 and time 1. So this means that for most of the CPG sites, the starting point, the set point is different, but the aging rate is exactly the same in people with Down syndrome and controls. We then wonder if there are at least a subset of CPG sites which has a different trend, so which show different aging rate in people with Down syndrome respect to control, with a different set point or also the same set point. We applied different and alternative statistical approaches to identify these probes, so in an interaction terms, or we analyzed specifically the longitudinal data set. Without going too much in details, I can tell you that there are very few CPG sites that show a different aging rate in people with Down syndrome respect to control. This is one of the most notable examples. You see this CPG site has a slight increase with aging, which however is significant in the controls, and in people with Down syndrome, you see that the aging rate, the hypermethylation rate during aging is higher. But I repeat, there are very few CPG sites that behave in this way. So to answer this second point, this second set of analysis, we can say that the epigenetic aging in people with Down syndrome has, in the vast majority of the cases, the same rate of the epigenetic age of euploid controls. What changed is the set point. So the epigenetic aging in people with Down syndrome is premature, but not accelerated. And this conclusion is supported by the analysis of the epigenetic clocks. You know that epigenetic clocks are algorithms, mathematical models, that take as input the DNA methylation value of a set of CPG sites, not too much, some hundreds, so not the entire microarray like we have analyzed, and return as output an estimate of the age of the individual, which is called the epigenetic age. And this epigenetic age can correspond to the chronological age, be higher or be lower. And in 2015, we published this paper in which we demonstrated that people with Down syndrome, this was basically the T0 of of the longitudinal course that I present you today. So we demonstrated that in this subset of people with Down syndrome, the epigenetic age was higher than their chronological age. But unfortunately, we used the term accelerated epigenetic aging in the title. And today we know that probably this, not probably, for sure, this title was not appropriate because the epigenetic aging of people with Down syndrome is premature, not accelerated. And here you see very well this thing. These are the results of the epigenetic clock on the longitudinal data set. So again, each line is an individual. And you see that, okay, people with Down syndrome have an epigenetic age which is always higher than their chronological age, which is higher than their chronological age, but the extent of the discrepancy between epigenetic age and chronological age is maintained constant during their life. So again, this proves that it's not accelerated. The last question is for sure the most interesting, but also the most difficult to be answered. So the question is there any relationship between the DNA methylation change that we observed so far and the phenotype of people with Down syndrome, also considering their variability also during aging? And here I can present you only preliminary results because we still have to conclude the analysis on the entire data set. What we have done so far is to identify those CPG sites whose DNA methylation is associated with the score of the test for severe impairment, which is a test, a neuropsychological test that can be used to detect dementia in people with Down syndrome. And this was the test that was available in both the Bologna and the Turin cohort. And what we found is that there is, again, a limited set of CPG sites which show an association with the score of TSI. And the interesting thing is that one of the CPG sites that we identified, this one, is exactly the one that I showed you before. So one of the few CPG sites that showed an accelerated epigenetic aging, let's say, in people with Down syndrome. And this CPG site maps in a gene which has been already involved, whose DNA methylation has been previously involved in Alzheimer's disease. So it will be interesting to confirm its association with dementia in people with Down syndrome. So to conclude, the analysis of this epigenetic data suggests that epigenetic aging is somehow intrinsic in the developmental defects at the basis of Down syndrome, and that in large part it's premature but not accelerated. And so it doesn't accumulate during the life of people with Down syndrome, with few exceptions, like the CPG site that I show you, that potentially, but I repeat, we have to confirm this data, can be associated with some phenotypical characteristics of people with Down syndrome. Just to comment these results, two considerations. First of all, consider that what I've presented you is somehow the normative aging of people with Down syndrome. So we found what changed with age basically in all people with Down syndrome. But this does not exclude that there are subgroups of people with Down syndrome that have different aging rates. And this, of course, should be investigated in next studies, also considering the potential effect of environmental factors. And the other point is that we presented the data of one of the hallmarks of aging, but of course there are all these other hallmarks of aging that can impact on the trajectories of aging of people with Down syndrome. So it will be interesting to analyze these other hallmarks, considering their evolution during the life of people with Down syndrome in order to understand if they are premature, accelerated, or both. And I conclude, I want to thank again the task force and all these colleagues and first-time colleagues, friends that work with us on this topic, and of course, all the participants with Down syndrome and their families that participated to this study. Thank you.
Thank you, Maria Giulia, for a very, very, very interesting talk. Are there any questions? Yes, I see. Okay.
Oops, sorry.
Lucio, you are asking whether you can put a question?
Yeah,
Lucio?
yes, please.
Sorry.
Please.
Yes.
Please.
So my question is whether your results are identical, whichever tissue do you use? Or is there...
Yes, this is blood. These are blood cells.
So the question is, what about if you use a different tissue? Is there a possibility that you find some organs that have, in which the methylation proceeds in a way that is different from others?
Okay, yes.
And if I can ask a second question related to this one, is whether this study can be done in an in vitro system, in tissue culture, say like iPS that are differentiating into neuronal cells. Would you see a change in methylation that could be, and I mean, would you expect to find a difference between trisomic and non-trisomic?
Okay, thank you. So regarding the first question, yes, we analyzed blood cells. In general, we know that the the different tissues of our body have different aging rates. For example, liver has a slower aging rate compared to the other tissues. So it's not necessary that what we found in blood is applied also to other tissues. I can anticipate you that I've tried to analyze also publicly available data sets from brain. And in brain, in the general population, we know quite well what are the DNA methylation changes during aging. And for example, we have demonstrated that these changes are accelerated in Alzheimer's disease. So I've tried to look at the existing data sets from people with Down syndrome on brain, and I saw that some of these CPG sites that change with aging in the general population change also in people with Down syndrome. So there are sites that get hypermethylated during aging in brain. They are more methylated in people with Down syndrome. But I was not able to calculate the trajectories, the slope, because there were too few points and with a small age range. So the data set was not adapted to perform this kind of analysis. Regarding the second question, I know that DNA methylation has been measured also in neuronal progenitor cells differentiated from iPS in people with deriving from trisomic cells, and they found large DNA methylation differences. I could not analyze this data, so I cannot tell you whether their epigenetic aging patterns are similar or different. Regarding the iPS itself, actually, I don't know. It's never been done. The iPS have a DNA methylation profile which is very similar to which is that of the reprogrammed cells. So I don't know what I can expect. I can tell you that in iPS the epigenetic clock is basically 0. So the age of iPS is 0, independently if you derive iPS from young or old people.
Thank you very much.
Okay, there is a question by Maria Liudino. What is the earlier age that you have compared Down syndrome and controls?
What is, sorry?
The earlier age. Imagine the younger sample.
Okay.
I don't know, the same
Yes.
thing.
In our data set, the younger point was 14 years, but most of the samples went from 23 years, let's say.
Okay. Then Emanuela Busi asks, which tissue did you analyze in your cohort? Sorry, I probably missed it. Did you confirm your results in other tissues?
I'm sorry, this is my fault.
Okay, okay. Right, same question. Okay. And then I do not see.
Dine, sei question.
No, sto scherzando.
I don't, not in the chat. Okay, here is Dan Nizetic question, please.
Hello.
I can hear some.
Can you hear me, Julia?
Yes.
Sorry?
Yes, Dan, please put your question, please.
First of all, congratulations. This is a really fantastic study. I really enjoyed hearing this and the way you approached and asked the questions. So congratulations, really great study. And really exciting. I'm very encouraged to see that you found some of the things similar to our study with IgG glycosylation, which
I know very well, yes, your study, and I noticed that exactly the pattern is very similar.
That they are parallel.
Yes.
So this is really interesting. It means that a lot of it happens in utero during development, and it sort of stays and doesn't change further. So we are very encouraged that the data support each other. What we also found, and this has to be investigated further, is that besides the methylation change, we see DNA repair related defect, which we have identified at least one of the genes, DYRK1A, might be participating in causing this, but the relationship between this and the phenomena you found needs to be investigated further.
For sure, yes, I agree, and also I know that because in the lab where I previous work, they investigated this topic, there is a relationship between DNA damage and establishment of DNA methylation patterns, and I totally agree that this potential link relationship should be further investigated.
Okay, thank you. Are there any other questions? No, I don't think so. Okay, thanks a lot, Maria Giulia. So we can move on to the third speaker of this first part of the symposium. The speaker is Silvia Lanfranchi from the University of Padova, and the title of her talk is Cognitive Profiles in Children and Adolescents with Down Syndrome. Please, Silvia, the floor is yours.
Thank you. Thank you. So I will try to share my presentation. This is always a stressful moment for a speaker.
Yes, that's true. Will it work or not?
Yes, because otherwise you don't know what to do.
Okay, we can see your screen. Yes.
Okay, I will put it presentation modalities.
Yes. Okay.
Okay, thank you.
Okay, perfect. Thank you.
Thank you. So, good afternoon to everybody. First of all, I would like to join the colleagues before me in thanking the Down Syndrome Task Force and Lucio Nich for organizing this very interesting conference and for giving me the opportunity to present you some ideas about cognitive profiles in children and adolescents with Down syndrome. All these studies that I'm going to present you today are a part of a broader project that we are conducting in collaboration with Professor Stripoli, Maria Chiara Pelleri, and their research group at the University of Bologna, and Chiara Locatelli, Giuseppe Ramaceri, and their group at the Sant'Orsola Hospital in Bologna on the relationship between genotype and phenotypes in Down syndrome. This broader study aims at looking at what are the factors that are correlated with intellectual disability in individuals with Down syndrome in order to better understand what can we do on the side of treatment in order to support them and to improve their cognitive functioning. My presentation today is more on the behavioral side of the story. So I will talk about the data on the cognitive and behavioral profile in individuals with Down syndrome. What we know so far about cognitive aspects in Down syndrome, well, the main thing is that almost all individuals with Down syndrome have intellectual disability, so a delayed intellectual development. Usually intellectual functioning is measured with the IQ, and we know that the variability in IQ is very high, ranging from 25 to 70 points of IQ with a mean of 50. And we also know that IQ is correlated with age, and specifically IQ tends to decrease with age in this population. However, this doesn't mean a loss of skill, but it is justified by the fact that the speed of development in Down syndrome is slower than in the typical population. And so the gap between the skills in individuals with Down syndrome and typically developing children increase. But the good news is that this is not a loss of skill, but individuals with Down syndrome through all the developmental years still continue to acquire new skills. Many studies in the past focused on language characteristics in Down syndrome, and they found that language could be an area of particular impairment in this population. And in particular, the productive aspects of language is more impaired respect to the comprehension, or more impaired respect to the non-verbal communication and language that are less impaired. Moreover, the aspects more impaired in language are grammar and morphosyntax, while the vocabulary seems to be more preserved. Other studies focused on visuospatial skills, and those studies show us that this could be an area that is relatively preserved, although it probably depends on the task that we use to assess visuospatial skills and on the subdomain explored, with some areas that are more preserved than others. Finally, talking about cognition in Down syndrome, we know that a particular difficulty is present in executive function, a set of higher order cognitive processes involved in planning action to perceive a goal. So far, in most of the cases, all those aspects have been considered separately. So we do have studies that focus on intelligence in Down syndrome, studies on language, studies on visuospatial skills. But we thought it would have been interesting to look at language, visuospatial skills, and intelligence together, because we know that language on one side and visuospatial skills shape intelligence. We do have a verbal aspects of intelligence that is involved when we make reasoning with words, and we also have a visuospatial aspects of intelligence when we manipulate object, real or mental. So in this study, that is a part of the doctoral project of Sara Onivello, we wanted to explore the cognitive profile of children and adolescents with Down syndrome, looking at the verbal component of the intelligence and the visuospatial component of the intelligence. Based on the previous study, the hypothesis will be that visuospatial intelligence could be somehow more developed than verbal intelligence. And this is because language is an area of particular impairment. So we thought that it might have an impact also on the verbal aspects of intelligence. To this purpose, we assessed a group of 72 children and adolescents with Down syndrome, aged between 7 and 16, using a Wechsler intelligence test. And this is the result. So looking at this first graph, I believe that the main result, the first result that we found is the high interindividual variability. You can see all the singular lines are each child, and the variability is very, very high. However, a mean profile in this group emerged, and interestingly, this profile is a flat profile with no differences between verbal intelligence and visuospatial intelligence. That is something different from what we would have expected. And this profile is confirmed also when we ran the analysis, not on the two main indexes, but on the specific subtest that we used. So the main result, as I said, is interindividual variability. And for example, this pool of researchers highlighted the importance of understanding interindividual variability in Down syndrome, since it is from double to triple respect to the typical population. And in order to understand the interindividual variability within our sample, we decided to run a cluster analysis in order to see whether it will be possible to identify subgroups of children with different characteristics. And in fact, we did found 3 different subgroups. The first subgroup had a profile similar to the whole group, with a similar level of verbal and nonverbal intelligence. It's interesting to note that this was the group with a higher level of intelligence compared to the other 2. Then we found a second group that has the profile that we expected, so with lower verbal intelligence and higher visuospatial intelligence. In this case, this group is the one that has the lowest level of global intelligence. And interestingly, we found also a third group that has exactly the opposite profile. So it has visuospatial abilities, visuospatial intelligence similar to that we found in the visuospatial group, but higher verbal intelligence. So it has exactly the opposite profile with better verbal intelligence than nonverbal intelligence. At this point, of course, the question is, how can we explain these different profiles? Of course, the first hypothesis could be age. So since there are also different levels of global cognitive function, we could expect that age explain the 3 different groups. But this is not the case, since when we compared the 3 groups for age, we couldn't find any different. So it's not age, chronological age, the aspect that explain those different 3 groups. So we should find other hypotheses, and probably we can try to explain this result using the words of the group of researchers that worked on inter-individual variability, highlighted inter-individual variability in Down syndrome. So probably it's looking at the cognitive profile just in terms of an extra copy of chromosome 21 is probably simplistic, but many other genetic and epigenetic and environmental factors probably play a role in determining how the Down syndrome phenotype expresses itself. And we believe that it's very important to better know and understand what are those factors and how those factors works and interacts with each other in order to determining the different phenotype. We can speculate, but it is just a speculation, that the visuospatial group is probably the profile more linked with genes. It is the profile that we would expect on the basis of what we know on Down syndrome characteristic. And of course, I am a psychologist, so for me, it's very important also to look and to think at the environmental aspects that may play a role. Probably it's possible to speculate that on the other 2 groups, environmental factor may have had a role in determining different profile. For example, I think about a stimulating family environment where parents are particularly sensitive to the language development of their children, and they are sensitive to stimulating the language development of their children. But also I think about early intervention. In the last year, we have understood the importance of early intervention on language, and that we don't have to wait that the language is developed in order to work on language, but we can start to work earlier, working on the precursor on language, fostering the precursor of language in order to help the language to develop better and sooner. And it's possible to hypothesize that those environmental factors may have had a role in determining the better verbal intelligence characteristics in the other two groups and the better global developmental level. But of course, more studies are needed in order to demonstrate those things or to see whether other aspects have an impact in determining these different three subgroups that are homogeneous in numerosity. Another question could be what happens at earlier ages? And usually in preschool age children, we don't assess intelligence per se, but we assess development. Development that, of course, is made by cognitive development, but also language and communication development, but also motor, gross motor and fine motor development, but also personal, social and emotional development that are all aspects that support the development of the intelligence in a child. And for this reason, in order to explore this question, we assessed a group of 43 children aged between 3 and 6 using the Griffith scale that are a test to assess the global development in the child. And this is the profile that emerged. Once again, the first result is the high inter-individual variability, and this variability is even higher in the language and communication domain respect to the other domains. Looking at the profile, we can see that the performance in cognitive development and in communication and language development are at the same level. They go together at this age. Moreover, that the personal, social, and emotional area is an area of strength in the profile of this group of children of preschool age. And finally, that gross motor skills are more developed respect to fine motor skills. Even in this case, the question could be, can we identify a subgroup of children with different profiles? And the answer is yes. We run also in this case a cluster analysis, and we could identify two different subgroups. The first one that had a lower global developmental level and an area of weakness in language and communication. And this was also the younger group. And then we found a second group with a higher level of development that was also the older group with an area of strength in personal, social and emotional skill. So to sum up so far, what is the take-home message is that the profile of the cognitive profile and developmental profile of children with Down syndrome is articulated, and it's very important to explore and to understand the articulation in the subdomains, but also that it's possible to identify more than one phenotype in Down syndrome, more than one subgroup with different characteristics. And this is particularly important also from the point of view of intervention. So probably on the cognitive aspects, there is not one type of intervention that works for everybody at the same way. And this is also because children with Down syndrome are not all the same, but they have different characteristics. So understanding the different characteristics can help us to give to each child the type of intervention that is better for him. In the last part of my talk, I would like to focus for a few minutes on executive function that are a set of higher order cognitive processes that are important for completing goals. For example, working memory, inhibition, planning, cognitive flexibility, or emotional regulation. We know that this is an area of particular impairment in the profile of individuals with Down syndrome. And so even in this case, we wanted to look at the sub-profile, comparing the different domains that compose the broader domain of executive function. And we wanted to look also on a developmental way, comparing the profile of children at the preschool age and children at the school age. We assessed the executive function through the brief. Brief is an indirect tool that assesses problems in executive function through parents' report of behavior that the child has in everyday life. And as we can see, the profile, first of all, the profile of executive function in both groups is not a flat profile, but has an area of strength and weaknesses. Moreover, it seems that on a global level, school-age children have more problems in executive function than preschool-age children. The profiles have some characteristics in common. For example, both groups had an area of strength in emotional control, and both groups showed an area of weakness in working memory. However, more weaknesses were found in the school-age children, also in shifting and in planning organization. Finally, we wanted to look at the correlation between executive function and adaptive behavior. Adaptive behavior are behavior that we normally use in everyday life for taking care of ourselves, taking care of the house, interacting with other people, and so that are that everyone use in everyday life, so they are fundamental for everyday life. In the preschool age children, we found a correlation between working memory and communication skill. But I believe that more interesting are the result of the school age group, where almost all the aspects of the executive function that we explore correlate with everyday life, and so suggesting that in particular from school age to approximately from 6 years of age, executive function become very important in supporting all the behavior that we use to function in everyday life. So from here, I believe that the challenge is finding the possibility to train early executive function in order to have a cascade effect on many other developmental domains and on many other behavior on everyday life. And that's why in the last 2 years, we worked on another project in collaboration with Deborah Fiedler at the Colorado State University in order to develop a program to foster executive function in preschool age children with Down syndrome. This is a phenotype-informed intervention that also uses a community-based approach. So in building the intervention, families were involved from the very beginning in order also to listen to their needs and their idea about the development of their children. So if you are interested in knowing more about this project, tomorrow one member of our team, Sarah Colayan, will present it. And for now, I have finished, but I would like to conclude with a big thank you to all the members of the Camminando per Mano Sindrome di Down lab group that worked on this project. So Sara Nivello, Francesca Pulina, Chiara Marcolina, and Sara Colaianni, and to all the Bologna's group, so Pierluigi Stripoli, Chiara Locatelli, Giuseppe Ramaceri, Maria Chiara Pelleri, Francesca Antonaros, Beatrice Vione, Alison Piovesana, and Maria Caracausi. And thanks to all of you for your attention. This is the website of our lab, so if you are interested, you can go there and look for the update of our project. Thank you very much for your attention.
And thank you for your talk. The discussion is now open. Let me see whether there are hands or someone in the chat. I don't see any question.
I see
Well,
one.
One
my
hand,
hand is a little
maybe.
bit dark. I have a
Sorry?
hand that is a little bit dark. Maybe I should change the color of my hand.
No, I can
May
see
I ask
one.
my question?
Yes, sure.
Okay. So the question is whether your data imply that there is a higher variability in the Down syndrome population with respect to the general population. So I wonder whether when you make three groups for subgroups for visual, spatial, and verbal intelligence, those are only found in Down syndrome population, or can you find a similar groups in the general population?
Well, I don't have a real, I don't have an answer because nobody did the same study with a cluster analysis on the typical population. I would expect that, yes, we could find the same subgroups also in the typical population. I would say that probably it will be more likely to expect the three subgroup in the normal population, because in the normal population, there are children that are more verbal, children that have a more visual intelligence, and children that are good in both. So I would expect the three groups in the typical population. I expected less the three groups in the Down syndrome population because the results of the previous literature says that language is an area of particular impairment. So I would have expected that this impairment in language would have had a role in determining lower verbal intelligence respect to visual spatial intelligence.
Thank you very much.
Other questions? No, I don't see any other question. I have a question to you and Julia, perhaps, because your hypothesis, a possibility is that epigenetic factors can account for the large variability among different children or people or adolescents. I'm asking to Julia at the same time, is it possible that such a big difference is accounted by epigenetic factors or not? What do you think?
I can give you an answer because one of the parameters that we try to analyze, but I repeat, they're only preliminary data, is IQ. And we exclude those participants that have dementia because, correct me if I'm wrong, but I think that IQ is not reliable in this population. And there are some genomic regions that have a significant association, but personally I was not really convinced by the association that I found, that is, they are statistical significant, but in my opinion, not always what is statistical significant is also biological significant. But this was exactly what we wanted to identify. So epigenetic basis of the variability in, for example, in this case, IQ in people with Down syndrome. I don't know if IQ of adults can be used as IQ of children. Maybe if we perform the same analysis in children, the data are less dirty, let's say, less affected, but by all the history of life of people, of children and adults with Down syndrome. And in children, we will be able to find more evident association. But Prof. Lafranchi can correct me if I'm wrong.
Sure. If I can add something, the difficulty in those studies is to bisect all the aspects because, of course, I believe that epigenetical factor will probably play a role in determining the different profile, but I believe also that environment also shapes what epigenetic factor do, and so the difficulty is to bisect and individuate the effect of epigenetic factor and environmental factor. And the more the child is old, the more environmental factor had time to shape the development. And so this is the difficulty of this kind of study. We are, of course, to do this kind of study, you also need a very big sample because you have to take into account many variables. And that's why we are trying to do a broader research group where also Maria Giulia is included, Maria Chiara Pelleri, to create a big data set on Down syndrome with many different genetic, cognitive, psychological variables in order to make this kind of studies. So hopefully, maybe for the future, we will have the possibility to run this kind of analysis.
If I can add, it will be interesting also to analyze during time if the groups that you have identified also maintains in adults, so the same clusters are maintained.
Sure. I think that it will be very important to run longitudinal studies. So ideally to start to follow a cohort of children very young and then follow them developmentally, because this is the only way to really to study the development, following the development at different stages. We try with cross-sectional studies, but of course, the better way to study development is longitudinally, following the same cohort longitudinally.
There is question of Filippo Caracci. Filippo, please.
Thank you, Renata. It's just a comment about Silvia that I want to thank you, and Maria Giulia answer to the question about the longitudinal approach, because my question was related to the different view that we have on the cognitive deficits along the time, because working memory in children is different from working memory deficit observed in an early phase of Alzheimer's disease in the same patient. The question is relevant because provided that we can consider different hypotheses for the pathophysiology of cognitive working memory deficit, including also, I don't know, for example, not only epigenetics, but also cannabinoid system and the other probably molecular mechanism underlying these deficits. The question is if working memory deficit is a trait, a cognitive deficit present along the life, no? It seems not so easy to study a new treatment, also a pharmacological treatment that can start in an early phase and then continue also in a late phase. So the question is, the question for you, Silvia, is how much is constant this working memory deficit?
It's
I know that it's a difficult question, but
it's quite constant. Of course, we have studies on working memory starting from 3 to 4 years of age, but because before this age, it's difficult. We don't have yet the tools to assess working memory. But the literature says us that from 3 years of age onwards, working memory is one of the area of particular weakness also within the domain of executive function. And there are a huge number of studies on working memory in Down syndrome, and almost all agree that there is a specific deficit in this area. More in the verbal aspect than in the non-verbal aspects, but still is an area of difficulty.
Okay, Lucio?
Yeah, just a very short comment, and they checked that I could change the color of my hand. So the comment was, let's not forget the genetic players, of course, because the real question there is how much the genetic plays a role with respect to epigenetic and so on. So that's all.
Sure, of course. This is very important. And as I said at the very beginning of my presentation, those are data taken from a broader project where also the genetic part is taken under consideration. And I believe that tomorrow Pierluigi Stripoli will give you the genetic part of the project, so you can put also the thing together.
Okay, thank you.
Okay, if there are not other questions, I think we can conclude this first part of our symposium. I wish to thank again all the speakers for their exciting talks and for having accepted to take part in this symposium. And the second part, we have a few minutes of coffee break. And the second part will start at 4 p.m. in a few minutes. So
Let's say 4:05, so we'll have 15 minutes, okay?
Yes.
4:05, 15
4 or 5
minutes.
minutes. Okay.
Okay.
Okay, thanks again. See you soon.
See you soon.
Sì,
Filippo, possiamo parlare un attimo in chat?
sì. Arrivo.
Okay.
Okay, Lucio, do you think we can
I think so, yes,
start?
we can start, yeah.
Okay,
Please.
so welcome back to everybody. So Renata and I would be pleased to present the second part of this international session. And we are pleased that after a few years, we succeeded in inviting and having the warm acceptance of very well-known scientists in the field of Down syndrome from the international community. And today we are very pleased to have two speakers, Professor Della and Professor Della Torre, hopefully, which are, I'm sure, very well-known scientists in the field. You all know them very well. But I'm very pleased to thank so much Jean Della because since we are approaching to the 10th anniversary of the International Society of our society, he has been the first president. So I would really like to thank, and I'm sure that next year we will celebrate together in Rome the 10th anniversary of the International Research Society. And I'm also very pleased to introduce Jean Della to all of you. Today, he will talk about data regarding DYRK1A, a well-known triplicated gene, which has been long studied for the paramount role in brain alteration in Down syndrome. In particular, today, Professor Della will highlight the complex role of DYRK1A in Alzheimer's disease. And in the context of Down syndrome, he will also discuss the link of DYRK1A as a triplicated gene for the early development of Alzheimer's disease in Down syndrome. So, Professor Della thank you so much for joining this session, and we are all pleased to hear your talk.
So thank you, Marzia, for this nice introduction. I have to share my screen.
Sure, as you know.
So is it okay?
Yes.
Perfect. You just go on the full presentation screen and we are fine.
I am on the full
just maybe just the full screen. I don't know if you.
Oh yes, you're right.
Okay, got it. Sure.
Yeah. I have still a problem.
Do you need any help, John?
Sorry. No, it's okay.
Okay.
So can you see my arrow?
Yes.
Yes. Also, you can use it to point. This is fine.
Okay, fine. I have to move something again. No, it's not that. Sorry.
You can test. Okay.
Yeah. Okay. So, I'm glad to receive this invitation and to have the opportunity to present this talk on the role of DYRK1A and particularly in Alzheimer's disease. The last part of this talk was done in Marie-Claude Potier's lab in Paris Brain Institute, and in collaboration with Saint-Anne Hospital and Dr. Sarrazin and Paris-Saclay University, Dr. Botlander, Elizabeth Head in the University of Kentucky when she was in Kentucky, and Ayerlot in Irvine. And it was founded by different agencies, CNRS, National Research Agency, JPND, European JPND, and the Fondation Jérôme Lejeune. So I will divide this talk in three parts. First, DYRK1A as a multifunctional protein. second, a gene dosage sensitive protein, and finally, the complex role of DIR-QNA during AD in diploid and individuals with trisomy 21. So going back to the end of last century, the mapping of critical region of Down syndrome, critical region for Down syndrome, was done by studying people with partial duplication. And so we could map various features of Down syndrome. And after that, there was mapping and cloning of DIR-QNA and finding that the autolog of Drosophila mini-brain on human is DIR-QNA, and that it maps on the critical region. And we could also run the full transcriptional map of this critical region. And finally, we got the genomic sequence of DIR-QNA by sequencing the whole human chromosome 21. It was collaborative work chaired by Hattori. DIR-QNA belongs to a group of 5 kinases, DIR-QNA B, DIR2, DIR3, and DIR4. And so it's like all kinases with an active site, and it's common to the 5 kinases. But the main differences are in the C-terminal part, and the C-terminal part maybe explains the difference in the function of these proteins. So as I said, DIR-QNA has multiple functions, and one of the reasons is that it is found in the cell, both in nucleus and in cytoplasm. And so in the nucleus, of course, you have the function involving splicing, the transcription of chromatin, and in the cytoplasm, the synaptic plasticity, the neuronal cytoskeleton, the axonal transport. And it's also involved in controlling apoptosis and cell signaling. As I said first, it's also a gene dosage sensitive protein, meaning that in humans, when there are duplication of chromosome 21 or partial duplication containing DIR-QNA, the 3 copies of DIR-QNA induce phenotypic alteration. Some of them are found in Down syndrome, and that's both developmental alteration and cognitive alterations. And on the contrary, when there is only 1 copy of DIR-QNA, there are also developmental and cognitive alterations, and it's a kind of autism, which is now known as MRD7 for mental retardation, autosomal dominant 7. And so mice models have been constructed to try to understand more about the consequence of these gene dosage errors. Mice model with 3 copies of DIR-QNA reproduce some phenotypes found in Down syndrome, neuronal phenotypes, cognitive phenotypes, and interestingly, these phenotypes can be rescued when using DIR-QNA inhibitors to decrease the level of DIR-QNA in the body, like epigallocatechin galate, lecithins, Dandy, Indy, etc. There are now 40 or more inhibitors for DIR-QNA. And there has been also a mice model with 1 copy of DIR-QNA that have been constructed, and they reproduce some phenotypes of autism and of this form of autism, which is MRD7. Yeah, it's just an example of what could be assessed and mice overexpressing diaquane. The MBAC transgenic diaquane was built in collaboration with Yanero, and this paper was done by Aurore Tomaso. In fact, when we have those 3 copies, and when we look on brain slices, we can assess the property of neurons. One of this property are the long-term potentiation. And you can see here in black the long-term potentiation for the wild-type animals, so with 2 copies of diaquane. And in white, you can see the long-term potentiation for the animal with 3 copies of diaquane. And this phenotype can be rescued when using a treatment with epigallocatechin galate. And finally, I will describe briefly the involvement of diaquane in Alzheimer's disease. So first, I will talk about brain, and second, what could be observed when we look at plasma. The hallmark pathologies for the diagnosis of Alzheimer's disease are well known. They are extracellular plaque deposits of beta amyloid peptide and neurofibrillary tangles of hyperphosphorylated tau. 3 kinases have been found involved in these two pathologies or pathological features: diaquane, CDK5, and GSK3 beta. And it has been observed that there is an increased level of diaquane after the beginning of Alzheimer's disease. And this increased level is associated with tangles. And after that, there has been many studies showing that increase of diaquane is associated with 3 molecular consequences. The first one is amyloid beta production. The first observation was made by Kimura in 2007, bridging beta amyloid production and tau phosphorylation with the overexpression of diaquane. There has been a recent observation showing also that there is a direct action of diaquane on the level of amyloid beta, because diaquane is suppressing neprilysin, which is a degrading enzyme of amyloid beta. And so when there is an increase of diaquane, there is a decrease of neprilysin and an increase of amyloid beta. Secondly, tau phosphorylation is modified by diaquane inhibitors. So there has been different observations with different inhibitors. For example, INDI, one of these inhibitors, reverse the aberrant tau phosphorylation. That was done in mice, and rescued the repress and fat signaling induced by diaquane overexpression. Another system is the use of microRNA. The overexpression of microRNA 26A5P could repress tau phosphorylation, and which is acting on the level of diaquane. In fact, this microRNA could repress tau phosphorylation and A beta accumulation. And that was observed in a model of Alzheimer's disease, which is the delta E9 model. And there has been also another observation made on the well-known on the 3X transgenic AD mice. That's another model with another inhibitor, the DIR219, given before the beginning of neurofibrillary tangle pathologies. And it was observed that this, in fact, this inhibition given before the FT pathology, delays the onset of AD-like pathology. And the third action of DIRC1A is that DIRC1A phosphorylates splicing factors, different splicing factors, and so can modify splicing of RNA, and so can modify the corresponding proteins. And there is an alternative splicing of exon 10 of the gene for tau, and this alternative splicing produces 3R tau or 4R tau, two different forms. And normally there is an equal level of 3R and 4R tau. And when there is a dysregulation of this process, it is sufficient to cause neurofibrillary degeneration. And this tau splicing can be modified using by DIRC1A inhibition. That was shown on the Down syndrome model, the TS65DN. And when they are treated with epigallocatechin galate, this treatment can rescue the disequilibrium between 3R tau and 4R tau. So there is we have tried to summarize this all this observation in this on this slide, showing that when there is an increase of brain DIRC1A, and that could appear in aging of diploid individuals in Alzheimer's disease or in Down syndrome with Alzheimer's disease. So the consequence of this increase of DIRC1A is an alteration of tau phosphorylation, of tau splicing, and of APP processing, and inducing neurofibrillary tangles and toxic oligomers, and so a loss of function, loss of neurons, and dementia. But it's not all, in fact. There has been different studies to try to find non-invasive plasma biomarkers to detect early Alzheimer's disease, and some of them are the ratio of beta 42 over 40, phospho-tau, neurofilament light chain, GFAP, and all these markers identify the first phases of Alzheimer's disease at mild cognitive impairment stage. But we ask, in fact, if we could use DIRC1A as a biomarker, and if it could be earlier than all those described biomarkers. So we study a cohort created in St. Anne Hospital with 25 controls and 26 individuals with Alzheimer's disease. And we studied their plasma, and we quantified DIRC1A with a commercial monoclonal antibody. And we found, and it was not in fact what we were expecting, we found that there is a decreased level of DIRC1A in plasma from patients with Alzheimer's disease in comparison to control. And there is no difference observed with DIRC1B, the protein from the same family. So it's very specific for DIRC1A. And we observe also this difference on cell lines derived from AD patients, lymphoblastoid cell lines, where we observe also a lower level of DIRC1A for patients with AD. We performed a second study with an improved methodology and with two novel antibodies that we created in collaboration with CEA in Saclay. And that was done on two different cohorts, the same as precedently, the Paris cohort and a cohort from Munich, from a hospital in Munich. And in this time, we assess the level of 3 different markers, DIR-1A, BDNF, and homocysteine in the plasma. You can see DIR-1A in red. So DIR-1A alone was already quite good with a sensitivity of 0.85. But with the 3 markers together, we had a very high sensitivity of 0.95. We perform a third study with, again, we try to improve the methodology using the MSD apparatus to quantify DIR-1A with the same 2 antibodies than before. And in that time, we used an interesting cohort, the INSIGHT cohort, which is a cohort of elderly cognitive complainers. And when we stratified the 300 individuals for their level of DIR-1A, for the level of amyloid in brain using PET imaging. So we could stratify individuals in 2 categories: those with low, those with low amyloid load and those with high amyloid load. Those with high amyloid load have a lower level of DIR-1A than those with low amyloid load. And finally, we ran a 4th study on 3 different cohorts: a cohort of individuals without established cognitive impairment, senior cohort, a cohort of individuals with Alzheimer's disease, and we had also individuals with other neurological diseases. And finally, cohorts of people with Down syndrome, with Alzheimer's disease or without Alzheimer's disease. And that time it was done with pre-plated DIR-1A antibody. So again, with an improvement of the methodology. So that's the demographic table. So first, we studied this senior cohort created with 96 cognitively healthy volunteers, age healthy volunteers, and they received imaging study, PET, to quantify again amyloid. And again, we found that those with high amyloid load have a lower level of DIR-1A than those with low amyloid load. We performed the same study on the different group of individuals that we have. The young controls, the controls that I have shown previously with low amyloid load and high amyloid load, and individuals with Alzheimer's disease and with 3 other neurodegenerative diseases. So that was hippocampal sclerosis, frontotemporal lobar degeneration, and progressive supranuclear palsy. And the difference was less obvious for this category. And when we perform a correlation between DIR-1A level and the PET signal, we have we found a very strong correlation. After that, we had the opportunity through collaboration with IRLOT, as I said, to study lymphoblastoid cell lines from control, Down syndrome, and Down syndrome NAD. And Alzheimer's disease. And you see here that the level of Down syndrome of diaquane in these cell lines is increased in cell from people with Down syndrome in comparison with control. But this difference is less between people with Down syndrome and Alzheimer's disease. And also there is a significant difference between the two groups, Down syndrome and Alzheimer's disease, with a lower level of diaquane. So the same observation that we found for diploid individuals. So we collaborated with Kentucky University, and we were able to receive plasma from 70 individuals with a longitudinal follow-up on 3 or 4 years, meaning that for each individual we had 2, 3, or 4 sampling. And the mean age was 38 for people with Down syndrome and 51 for people with Down syndrome and Alzheimer's disease. And we found a very strong, significant difference between, for the level of diaquane, between people with Down syndrome and people with Down syndrome and Alzheimer's disease. So here we just combine the level of diaquane in the different category, and we calculated the ratio in the different groups. So we have this increase of the level of diaquane with aging. And also when we compare the people with Down syndrome with the age cognitive ALC, it's appropriate because, of course, you know that there is an accelerated aging in Down syndrome. You see that we found a quite coherent level ratio, which is not far from the 1.5 ratio. But when we perform the comparison between Down syndrome and young individuals, you see that the ratio is not 1.5, it's 4.4. So it explains that in the literature, sometimes you have a very strong difference not corresponding to the gene dosage effect. So our hypothesis at the present time is that an elevated level of diaquane in periphery during aging has a protective effect. So it's a bit strange, of course, because we have said that in the brain it could be a negative effect. In fact, we observe a similar situation in mice with hyperhomocysteinemia. In hyperhomocysteinemia, so that's mice that are mutated for cystathionine beta synthase. High homocysteine is associated with low diaquane in liver and high diaquane in brain. So it's contrary, in fact. And when we correct the level of diaquane in liver using an adenovirus diaquane to target the liver, we correct the diaquane deficit in liver, we correct the level of homocysteine in plasma, and we correct the level of diaquane in brain, and so it's a decrease in that case of diaquane. So this correction in periphery could have different effects in periphery and in the brain. And finally, I wanted to mention also this study in centenarians, where the authors have found, it's a very recent study, have found very weak correlation between neuropathology and neuropsychology. They have found a category of centenarians with plaques and tangles in the brain and no cognitive impairment. So the authors propose that there are resilience factors that have to be found. So we have just made this schematic view to summarize what we found in plasma. When we look at the level of diaquane in plasma, so there is an increase during aging which is higher in people with low amyloid load. And there is no increase in people with Alzheimer's disease. Clearly, what we would need is to have more data for this critical period in between those two states with low DIR1A and high DIR1A. And that for that, we would need a larger cohort, of course. To conclude, I would say that maybe there are ways, it could be optimistic, but there are ways to act on the level of plasma DIR1A. For example, by lowering the homocysteine level. And there are ways to lower this homocysteine level, not using adenovirus, but for example, a simple Mediterranean diet reduce the level of homocysteine. And the second point that I wanted to say is that we have to be cautious when using DIR1A inhibitors to treat Alzheimer's disease very early, because in fact that could have different consequences, opposite consequences in plasma and in brain. And of course, one solution would be to target brain for DIR1A with DIR1A inhibitors. Thank you for your attention.
Thank you, Jean, for your very comprehensive and outstanding presentation. So let's open the discussion, and I ask also Renata just to coordinate with me in case you see from the chat or hands up for questions. Yeah, just because I'm not in the right screen.
Dan?
Oh yeah, there is the first question. Sorry, Lucio. And then I see the first from the chat from Mara Dirsen. And what about the levels of DIR1A in CSF? Have you any evidence or measures of DIR1A levels in CSF?
We have not. In fact, we don't have any results in CSF.
Okay. Okay. Sure. Because.
But
so,
of course, that could be very interesting to do that. Yes.
Okay, so I'm just following also Dan and then Lucio and then Iris. Okay, let's follow this order.
Hi, Jean. Thanks for this very interesting and very thought-provoking talk. It's opening new directions, I think. So my question to you is, DIR1A is not known as a secreted protein. It's known as a protein that's, from all we know about it, is intracellular and in particular intranuclear, but also sometimes in other parts of the cell. So how do you explain its level in plasma? And my second question is, when you compare the absolute levels in concentration in plasma compared to, for example, if you mash up an organ, a brain or any other tissue where the DIR1A is expressed, what are the absolute differences between those two situations?
You mean when we have not done that in people with Alzheimer's disease, in fact? No.
In general, if you take just the.
Oh, you mean the amount in brain and in plasma?
Or any other tissue.
That we don't know.
Versus the plasma level.
No, that could be interesting, yes, to do that. For the presence of DIR1A in plasma, we don't know exactly the route, but you know, when we people have looked at various proteins in plasma, and they found a lot. Yeah, they found a lot of proteins.
May I?
Yes, Lucio.
I guess the activity of of DYRK1A is related to its kinase activity. Is that correct? If it is so, you are measuring amount. Does the amount correlate to the activity, to the kinase activity?
Oh, you mean in plasma? We are.
Yeah, even in plasma.
Yeah. Well,
yeah.
we have not done that. Yes, that could be interesting. Yes, to do the activity in plasma. But what we have tried is to, yeah, I think we have tried to do that. Yes, it was not changed. In fact, no, in plasma it's not changed.
Okay, next question from Iris. Would you suggest DYRK1A inhibitors in children and avoid them eventually in the elderly as an approach? What do you think?
Well, as I said, the possibility is to use methods to target the brain with inhibitors in elderly.
Okay. Question from Jacqueline London. Did you have the opportunity to assay all Down syndrome persons who do not exhibit Alzheimer's future? Because not all persons with DS have AD pathology.
So we had, in fact, two different cohorts, and we compare people with Down syndrome and people with Down syndrome and Alzheimer's disease. Of course, it is the future situation of people with Down syndrome. We could not have all the ages, and it's of course those studies have to be extended and confirmed with larger cohorts.
Okay, Simona?
Yes. Okay. Thank you, Jean. I would like to understand better the interplay between DYRK1A and the homocysteine in terms of biomarkers, because for me, probably I miss, but there is a gap. Why? Because homocysteine is a marker for inflammation. So what do you think that the presence of this high level of homocysteine you can correlate with the neuroinflammation, or what do you think we can use these different markers together to also for prognosis?
Well, in fact, it seems that the role of DYRK1A is, or it has been said so, that it is more or less pro-inflammatory, in fact. But I don't think that has been done really in different organs and different conditions.
Okay.
Okay, there are other comments, maybe first from Pablo Elguera, maybe related to DYRK1A activity, because he's wondering if you have checked targets of DYRK1A in plasma, maybe just as an indirect measurement of the activity. I guess the meaning of Pablo's questions.
In fact, we have just performed a test with a peptide target of DYRK1A. It was not proteins per se, but it was peptide.
Okay. And then there is another comment of Mara. We don't know how levels in plasma are related to the brain levels until we don't get CFF, even if, of course, it is difficult, and maybe it's better. No, she suggests, if I'm correct, to avoid inhibitors in the elderly.
Oh, yeah. But in fact, well, if CSF is not brain, it could give an indication, but
Sure. Okay.
But of course, it's interesting to have the level of DYRK1A in CSF. It would be interesting, yes.
And the last question, maybe from Mara, if have you looked at the SAD mice, the humanized APP models, whether there is a correlation of brain plasma levels as we are discussing?
Not yet.
Okay. Okay, so these are the questions from the chat. Any other hands up before I thank Jean Delabarre for your very great talk, and again for being today with us and with the rest of the community. Thank you, Jean.
Thank you.
And yes, we are ready to introduce the second speaker. And let me give you a special thank to Dr. Della Torre today for being with us. So we are impressed by your presence today, and we are very pleased to hear your talk about, you know, new pharmacological approaches, as you are involved in a very big European project with other Italian colleagues. Called HiCode, Improving Cognition in Down Syndrome. So I'm sure that all the community today knows most of the approach and maybe some of, you know, the preliminary studies on this project. And we will be very pleased to understand and to know more about the new therapeutic approaches for treating cognitive deficit in Down syndrome. So we want to thank and give you the podium, and we are very pleased to listen to your presentation. Thank you so much, Rafael.
Thank you. I can share my screen, hopefully. Sure. Wait. Here we are.
Yes, maybe just go in the full screen. Okay.
It's fine. Ok. Adesso Marzia e Renata, grazie per l'invito a partecipare a questo simposio. My name is Rafael Della Torre. I work in the Neuroscience Research Program at this Hospital del Mar Research Institute. And this afternoon, I'm supposed to talk to you about therapeutic opportunities in the treatment of cognitive impairment in Down syndrome. Okay. If you look at this picture that I like very much, that was produced by the scientists from Roche. It's very nice because, in fact, it shows that, as you know, Down syndrome, because of trisomy, has a lot of comorbidities related to cardiac function, endocrine function, and even some areas of the central nervous system. And for most of these disorders or comorbidities, we have, let's say, a drug, a therapeutic drug that we can, that allows us to treat these comorbidities. But where we are always failing is in providing a treatment to enhance cognition. And this is really the challenge of my presentation today, but it's the challenge of all you that are attending this presentation. I mean, our healthcare system is very efficacious in improving many of the comorbidities of Down syndrome, except cognitive performance. The idea is that what we want is that we want people that at present are dependent living to become independent living. And I think that over these last 50 years, we are closer and closer to succeed in this approach of independent living with managers. And we have really recently reviewing what happened in the last 10 years regarding clinical trials trying to improve cognition in subjects with Down syndrome. And we detect about 30 clinical trials in these 10 years, most of them concerning the use of pharmacological agents, but also there are some related to biological means or the device mediated. On your right side, you have the level, I would say, of evidence of what we know about many of these substances we have been trying. Most of them are just at the level of pilot studies. Some of them has been in the phase 1 that just shows you that the drug is safe, full stop. The main interesting things are the substances that have been assayed in the phase 2 and phase 3, where you are looking really at the efficacy of the treatments. As you can see, the number of drugs achieving this phase 2, phase 3 is lower. It's going lower and lower. If we look also at what happens in the preclinical studies regarding trying to find new therapies for improving cognition, Down syndrome, the landscape is more or less the same. I mean, we have a lot of studies targeting new pharmacological agents, and then some biological ones, and then there are some other approaches like behavioral approaches, dietary ones, multimodal approach, and even surgical, and also the number of let's say models that we are trying has been expanding from the trisomic mice to IPSSL, cerefish, and other rats and other models to investigate these new developments. The thing is that one of the underlying mechanisms of this cognitive impairment in people with Down syndrome is neuroinflammation. There are many factors, environmental factors, that healthy diet, reduced exercise, and also some comorbidities like hypothyroidism or diabetes or some autoimmune diseases that contributes to this situation. And this also opens the potential of investigating not only drugs that in the past were focused mainly on neurotransmission systems, but also to approaches, maybe therapeutic approaches that, I don't know, for example, the multimodal interventions that try to target different lifestyle factors of people with Down syndrome, that if you are able to modify them, they should result in an improvement in cognition of these subjects. And here we have what we call the therapeutic armamentarium of Down syndrome. We can start with symptomatic treatment, then we have trying to some disease modifying therapies or approaches. Then we have these multimodal approaches that they try to modify lifestyle factors that are also contributing to the cognitive impairment, immunotherapy, and even neurotherapy using some devices of electrical neurostimulation and even nanomedicine. The idea now is that we are going to review very quickly what happened in these 10 years and to understand where we are. One of the reasons why I understand that Martin and Renata invite me to participate this afternoon in this presentation is because initially with Mara, we were involved in the clinical development of epigallocatechin gallate, which is, as you know, a green tea compound that it's an inhibitor of D1A that Jean has been discussing previously. And the idea was that this ECG should, as inhibitor of D1A, that was one of the genes that was overexpressed in subjects with Down syndrome, should result in improvement in cognitive performance in these subjects. And Mara Diaz, particularly, and also John de la Barre, they contribute to these preclinical studies with epigallocatechin gallate and trisomic models, mice. And then we performed several studies. First of all, in adults with Down syndrome, these were just phase 1 studies to make sure that this compound was supposed to be safe. We have some indices of efficacy, and then we moved to this a larger study with 89 subjects known as TESDAT, where we tested the epigallocatechin gallate in adults. And we further performed a study in pediatric population known as Perseus study. Okay. And we did the full development of this the compound, although the results were showing that there was an increase on functional connectivity and some degree of normalization of cortical excitability and some improvements in episodic memory and executive functioning tasks. Globally, the effects of green tea as a cognitive, let's say, cognitive enhancer were quite good, not very, let's say, strong. It's also true that we have to demonstrate the performance of this EZCG combined with cognitive training in people that participate in the TESDA study against people that receive placebo but receive cognitive training. I mean, there was not let's say, the best situation where to demonstrate the efficacy of ECG, but of course, from a methodological point of view, that was the correct approach. We performed this ECG study in a pediatric population that, in fact, what it demonstrated this study in pediatric population essentially is the difficulty of performance these kind of studies in these populations, because not always the tools that we have to explore and to evaluate not only efficacy but other, let's say, the functionalities like I don't know, neuropsychiatric symptoms or functionality that works in young adults are really working in pediatric population, and this is something that is a field that really you have to work more extensively if you want really to do studies in pediatric population with Down syndrome. The thing is that in parallel to this study of ECG is today used for many families, and some of them they report improvements in some functional areas, particularly in the day-to-day activities of subjects, but essentially because it's a natural compound, there are no, let's say, pharma companies behind this compound interested in developing it. Then ECG on its own is at that end in terms that is a natural product that is not given to receive further support for its development. But in parallel to these studies with ECG, this French laboratory, Manros Therapeutics, has been working on synthetic DIL1A inhibitors, and they obtained, let's say, relatively good results in some cognitive tests, and to my knowledge, they are ready to start phase 1 studies to demonstrate at least initially the safety of these molecules, and in further studies, they will try to demonstrate the efficacy of using specific inhibitors of DIL1A. In practice, what happened in the last 10 years is that the studies focusing on the pharmacological treatment of Down syndrome has been initially trying to target subneurotransmission system. Why? Because it is true that in Down syndrome, several neurotransmission systems, here you have a list of cholinergic, noradrenergic, GABAergic, noradrenergic, and serotonergic, look or seems to be implicated in the pathophysiology of cognitive dysfunction. And then the idea is that, we are trying to treat these functionalities in these neurotransmission systems. These essentially concern the GABAergic and the cholinergic and glutamatergic system. My point of view and the point of view of others is that from our point of view, is that it is very difficult to see efficacy in the central nervous system diseases with molecules that target a single receptor, and that probably molecules that are more comprehensive in targeting multiple systems they have more chances to show efficacy, although there is a risk of sensitization and tolerance. But the thing is that we invest probably in the GABAergic system is where we invested most, let's say, resources, particularly because Hoffman LaRoche trying to develop a molecule, masmisanil, that was targeting the GABAergic system because it appeared that in Down syndrome there is an excess of inhibition by the GABAergic system, and the masmisanil trying to compensate in some way this deficit or the lack of balance between the GABAergic system and the glutamatergic system that you see in your picture on the right side. Well, the thing is that here you have masmisanil, which is the molecule that was developed by Hoffman LaRoche. You have pentylene tetrathol, and also we have bumetanide, and all these studies, either they fail or we don't have results. That in practice means that probably nothing happened, because if results were positive, for sure that they were the first ones to be to know them. Then globally, this GABAergic hypothesis that compensating this excessive inhibition of the GABAergic system in people with Down syndrome will improve cognition, fail, although there are some people that are still working on this hypothesis. I mean, it's not really a dead end, but still difficult way. The alternative was to use to try, because as you know, in the trisomy 21, one of the genes that is triplicate is APP, and APP, as you know, is associated with Alzheimer's disease and the accumulation of amyloid plaques in the brain in adults with Down syndrome. And then we decide to try different medications that, in fact, in practice, were designated initially for the treatment of Alzheimer's disease in subjects not with Down syndrome, for the improvement of cognition in people with Down syndrome. And we tried memantine, racemine, and SBL that target glutamatergic and cholinergic signaling. All these studies fail. Also, we tried inositol, fail, and also we don't have the data, but there are some a vaccine that is targeting some of the amyloid plaques that is ongoing. The only good news, in addition to maybe what has been reported previously from the Labar, is that apparently, at least for this period of the life of people with Down syndrome, that they start experiencing symptoms of dementia. There we have some biomarkers, and particularly the neurofilament light chain that appears to be a good biomarker for the early detection of Alzheimer's disease in people with Down syndrome. That could be useful in future clinical trials, for example. So what we up to now, we see that the GABAergic system failed, the cholinergic and the glutamatergic system also failed. Other hypotheses that has been tested have to do with the interaction of thyroid hormones and cognition, because in a study that was conducted by the Jérôme Lejeune Foundation, it appeared that there was some improvement in cognition in subjects that were treated with thyroxine. And then there was the hypothesis that maybe these newborns that were treated with folinic acid and thyroxine will improve cognition over time. And the thing is that we have been investing a lot of resources to this study combining folinic acid and L-thyroxine. And in fact, the results doesn't really support that this combination improve the development of young children with Down syndrome, and either they are not synergic. Some other alternative hypotheses that today are ongoing have to do, for example, on the alterations that are seen in Down syndrome regarding the mitochondrial function, and this had been related to the high expression of this cystathionine beta synthetase, the CBS, that is responsible for an overproduction of a gaseous transmitter, which is iodine sulfide, that may be related to this mitochondrial dysfunction. And then, of course, if you are able to correct this overproduction of hydrogen sulfide, you should improve in some way cognition. And this is something that is now today investigated for some researchers. Also the interaction with this cystathionine beta synthetase and liquane is something that is an aspect that also deserves some attention. The most recent hypothesis has to do with the involvement of the endocannabinoid system in cognition. Some results produced by my colleague Andrés Ozaita at the University Pompeu Fabra here in Barcelona. They were already showing that the expression of the C1 receptor in trisomic mice was much higher than the ones that you find on the wild type and in the wild type mice. And this is related to results that we generated in my laboratory, where plasma concentrations of 2-HG and IAA, two of the main endocannabinoids of in humans, well, in general in the endocannabinoid system, but particularly in humans, they were higher in people with Down syndrome as compared with the population that it has a neurotypical development. Then globally, there is let's say the hypothesis that there is an hyperactivity of the endocannabinoid system. And in fact, my colleague Andrés Ozaita also tried to demonstrate that by treating these trisomic mice, both trisomic mice model and the trisomic mouse expressing 3 copies of D1A, the transgenic mice, the TZD1A, they try to demonstrate that using an inhibitor of this endocannabinoid system, we could improve cognition. And it's true that using rimonabant, which, as you know, is an antagonist of the cannabinoid system, the mice treated both in the trisomic mice and also the transgenic mice, both when treated with this inhibitor of the endocannabinoid system, there was an improvement of cognition that was similar, let's say, of the performance of the wild type models. The problem, as you know, is that rimonabant is a molecule that cannot be used in clinics because some side effects, particularly related to the mental health area, depression and suicide tentatives, and then I mean, it's fine to use it in animal models. We cannot use in clinics. But and then what happened is that French researchers at the Magendie Center in Bordeaux, what they observe is that when there is an hyperactivity, particularly an excess of activity of endocannabinoids because of a very high dose of endocannabinoids, what happened is that they observed that there was some kind of compensating mechanism of this hyperactivity by increasing the concentrations of pregnenolone by 1,000%. And this increase in pregnenolone was able to mitigate this hyperactivation of the endocannabinoid system. And this was interesting because this pregnenolone was just acting in part of the functionalities of this receptor system. Instead of blocking completely, fully blocking the receptor, as it happens with rimonabant, it only blocks partially some of the functionalities of this receptor. And this was the basis to develop what is called today a new pharmacological class, known as the CB1 signaling specific inhibitors. Okay, that in some way what they are trying is to mimic this natural mechanism of defense against an hyperactivation of the endocannabinoid system mediated by pregnenolone. And there are several drugs today developed, and in particular, there is one that today is under clinical development, and is this one that we are testing in the ICOD project, which is a funded project by the Horizon 2020 European Union program for research. And today we already finished all the clinical studies in healthy volunteers. That means the single ascending dose, the multiple ascending dose, even the food drug interaction, and all these studies are completely clean. I mean, there are no adverse effects, and everything is fine. And we are currently doing the first studies in people with Down syndrome. At present, now we are just trying to make sure that the compound is safe and well tolerated in people with Down syndrome. But we have some tools that we believe that they are going to allow us to, in some way, to detect some improvement in cognition. I mean, and this will be the first step for the performance next year of phase 2 study to demonstrate the efficacy of the treatment. Nevertheless, because even for this CB1 selective antagonist of the endocannabinoid system, we have to demonstrate the efficacy. Then globally, if you look, most of our approaches, therapeutic approaches to treat and to improve cognition in Down syndrome fail. It's a disaster, frankly speaking. I mean, if you take into account the amount of resources, not only the economical ones, but even the intellectual ones we invest, that is a fiasco, full stop. There are many reasons, and I like very much this article that was published in Science in Molecular Medicine in 2020 that was looking a bit at the challenges and opportunities for translational therapies to improve cognition in Down syndrome. And it's for many, I mean, the problem is that he has not the solution for the challenges, but the challenges are well identified. I mean, we have many times a lot of problems in the translation of preclinical models to human ones. We have problems to recruit patients, to have well-powered studies to demonstrate the efficacy, but also we have problems to have let's say the right endpoint to demonstrate that this compound is efficacious, because of course, one of the questions is, what does it mean improvement cognition Down syndrome? I mean, it's something that looks very precise, but in fact it is a very broad concept. And the thing is that, as you say, every time we try new compounds that apparently it seems they work in animal models, when we move to clinical trials, they fail. And this is what happened to our colleagues in the Roche company, that they invest a huge amount of money, and afterwards we were unable to demonstrate any indice of efficacy of this compound. In fact, placebo even was better than the drug we were administering. And this was really very disappointing, because Roche really took very seriously the clinical development of this drug, and they try to do their best. Unluckily, results were not supporting the, let's say, the hypothesis, and I would say even the results in preclinical studies in animal models. Then, as I told you, one of the main obstacles of demonstrating the efficacy of the treatments we are developing in human clinical trials is the lack of generally accepted endpoints to assess the improvement in function in individuals with intellectual disabilities. And it is true that I'm always saying that, and I know that I will be killed by the professionals on this area, but there are as many solutions as neuropsychologists are today available in the market. I mean, everybody has his own test, neuropsychological test or battery of tests or something like that. And to explore people with Down syndrome, you have to find a way of in half an hour to be able to explore cognition and really to achieve a conclusion. And this is one of the undertakings of the national institutes in the States, and with the collaboration of the UC Davis and the Mind Institute. They took the initiative of developing what is called the National the Health Toolbox Cognitive Battery for intellectual disabilities. And this, in fact, it was not an initiative from people from, let's say, the world of clinical trials on Down syndrome, but from the people working in the field of Fragile X syndrome clinical trials, that there had been, I would say, before we identified 30 clinical trials for subjects with Down syndrome that developed in the last 10 years. The number of Fragile X syndrome clinical trials is much higher theoretically, because it's a genetic disease that you have just one gene as compared with a full chromosome. And every time these clinical trials fail, but sometimes the clinicians were very frustrated because the perception of patients or even some of the physicians participating in these clinical trials was, let's say, much better than the final conclusion taken by the outcomes that were pre-identified before starting the clinical trial to demonstrate efficacy. Then they decided to develop something new, a new score, and this is what is called NIH Toolbox Cognitive Battery for Intellectual Disabilities. That means it's used for Down syndrome, but can be used also for Fragile X. And they have two scores. One score that has to do with fluid cognition, and the other one with crystallized cognition. This, of course, has been validated and published, and is essentially the initiative of David Hessel from the Mind Institute that has been leading these studies. And the good thing also is that because you have a composite score on fluid cognition or crystalline cognition, agencies are, let's say, more ready to accept your results. Because the problem is that you go to the FDA, to the European Medicines Agency, and say, look, my molecule works. The problem is that you cannot say that based on 1 or 2 tests over 10 that apparently they were that significantly changed. But they want these regulatory agencies to look at a composite score that's saying globally, yes, the cognition of these subjects has been improved. Afterwards, you can do as many sub-analyses on which of the members of this composite score they were the ones that really mainly changed as compared with the other ones. But the agencies want to see a global score, and this is the good thing of this approach of this NIH Toolbox Cognitive Battery Intellectual Disabilities. Future developments. Probably neuromodulation will play a role in the treatment of and the improvement of cognition in people with Down syndrome. there are some ongoing studies using this transcranial direct current stimulation. We have some preliminary studies performing in adults and in adults with Down syndrome, and probably there is some hope that this approach is going to help. I mean, it's not going to be the solution, but it's going to help in the improvement of cognitive performance in people with Down syndrome. Our view is that probably the final treatment is not going to be a magic pill, but probably we need, of course, some compounds that are able in some way to improve cognition, but also we have to combine this with some lifestyle factors like diet, physical activity, cognitive training also, and why not also neuromodulation. Probably the PAC is the one that in the future is going to give us an answer how to improve cognitive performance in Down syndrome. E adesso vi ringrazio per l'attenzione per partecipare in questa presentazione oggi. Grazie.
Grazie, Rafael. This was really a very comprehensive and very full of information, criticism, inputs for discussion. So I'm sure we will have a lot of questions from the chat, from the people. Thank you so much. So again, questions on the chat or hands up? From the chat, sorry. Okay, to check.
Yes, there is a comment
maybe
from
the first
Mara.
one is, yes, the first comment is from Mara. And I read Mara's comment. We need to change the way we do clinical trials. Down syndrome population is too variable, and this cannot be overcome to achieve statistical significance or clinical relevance of the effects. It is great to have more tools, but we know by experience that what we measure in a clinical interview does not necessarily reflect what the person can do. It would be better to have longitudinal cognitive evaluation tools. And also Mara added that regarding GABA targeted treatments, the clinical failure is maybe also due to targeting only the postsynaptic component of the circuit, as the presynaptic elements are also altered.
No, well, regarding to the first comment, I think that our colleagues from London, they produced 2 or 3 years ago a paper that I like it very much. But in some way, in summary, what they say is that we have the perception that because people with Down syndrome, they have the trisomy 21, all of them, they are exactly the same. I mean, they are missing, well, we have an extra copy of chromosome 21, and all of them, they should be treated equally. And they say that no. The people with Down syndrome are extremely heterogeneous, and we have a lot of, let's say, of phenotypes, and our approach to see people with Down syndrome has to be that we have an heterogeneous population and not an homogeneous one. And I think this is correct. And in practice, the solution is to try to take into account all these variables when you do your analysis of data, I would say, as a confounding factors or as others to modulate the statistical analysis. Yeah.
Filippo, do you want to add any comments since I saw you on the chat, if you want to say some words about or...
The comment is shared with Rafael many times, the problem that to describe this complexity to regulatory agencies, because Down syndrome families and associations know very well this complexity. I'm not sure that the same approach is from regulatory agencies. So the first step is as Rafael said, and I would like to thank you again for his presence today, the validation of a new tool, and I hope it can be a common tool for different studies and different targets. It's just a comment, just to say thank you again.
There is another comment from Paco Tejedor that wants to talk with this very interesting talk. And he said that the initial hold pharmacological studies indicated that the EGCG cannot cross the blood-brain barrier. Has this been confirmed? And if so, which could be the active compound from green tea extract producing positive effects?
That's not true, because our colleagues from the University of Paris, they already published a manuscript maybe 2 years ago showing that EGCG was crossing the blood-brain barrier. I mean, it's just a matter of reading this paper and you have the evidence that this is correct.
Laura, just to clarify, the bumetanide trial is currently recruiting. It is not failed yet. So we see and we know the very active community of science.
I'm sorry, I couldn't find the...
Laura? Yes, you. Oh, sorry. Okay, you can talk.
I couldn't find the tool to raise their hand, but
that's fine. You can go.
No, yeah, it's not failed yet. We're recruiting, so we don't know.
No, I agree with you. I didn't say there was fail. However, there was a fail in other molecules, not in your one, in bumetanide. The problem is that we still for many years waiting for results, but and we'll continue to wait. I mean,
to get all the approvals. We are
sure,
recruiting.
I imagine, yeah.
Sorry, is it possible to make a question?
Yes, sure.
Okay. When you talked about the efficacy of different molecules on the cognitive efficiency in Down syndrome, and you told us that several molecules failed. I ask just a clarification about this. That is, the less of effectiveness was on a particular set of cognitive abilities, or was it in the general cognitive ability, let's say an IQ? So because every one of us know that cognition is not a number, but cognition is a vast array of different abilities. So I think that one of the problems may also be on what part, on what sector that specific molecules can act. So I don't know if this question is correctly posed, but I talk as a clinician. My curiosity is about the match between the complexity of cognitive abilities, of cognition, and the effect of a single molecule.
I mean, for many molecules, that the ones, for example, that were used in the past for the treatment of symptomatic treatment of Alzheimer's disease, they follow an approach which is very similar to the Alzheimer's disease trials. And the problem is that they couldn't report any improvement that was, let's say, acceptable, but by the regulatory agencies, because these are the ones that at the end they decide. But I would say that our perspective in Barcelona at least is that doesn't matter which scale do you use or battery you're using for in measuring cognitive performance. If there is not a functional improvement, I mean, to have something that changed maybe statistically in one scale 0.5, if there is not improvement in functionality, I mean, for the family, this is useless. Then for us, the important thing is not really to have an improvement in cognition, of course, but this improvement in cognition should be combined by an improvement in functionality. If you are not able to demonstrate that, our clinical trial failed, really. Doesn't matter if some tests passed the statistical screening and the other ones not. I mean, if the families are unable to notice that there is an improvement in their children or their peanuts, you are wasting time and money.
Okay, sorry, just very quickly. I don't know the literature about this, but you know, can you tell me if the adaptive functioning was measured when using these molecules besides the cognitive abilities? That is, if somebody used measures like Vineland scales or something that measured the adaptive ability, the capability of people, of persons to function better in society or in relationships.
In many studies, we are using these measures, but as I told you, the issue and these I agree, not always is like that, but I agree with the regulatory agencies. What they want to see, the regulatory agencies, are a composite score, somebody that combines different cognitive areas. And the combination of these cognitive areas says, yes, your treatment is, let's say, better than placebo. The second question is, that is a big question, is what is statistically significant and what is clinically relevant? Because in your study, you can demonstrate some, even using these composite scores, that there is an improvement of your treatment of a placebo, but you say, but it's not clinically relevant. And then we come back to what I was saying to you, that yes, maybe we can demonstrate some improvement in cognition in some areas, but there is not improving in functionality. And this is why the regulatory agencies say, no, enough is enough. We want to see a composite score. Show me that this is working. Tell me what means a clinical significant change, and then do your clinical trial, and then show me that really this clinical trial is producing results that demonstrate that your change is clinically relevant. That's today the main issue, and which is not easy, I would say, but it is the main issue today.
Okay, I agree with you that there are so many issues that it is not so easy to measure what the effects of a certain molecules can be on cognition, especially in Down syndrome.
Yeah.
Lucio, maybe?
Yeah, just profit of the presence of other experts on DIRC1A. I figure out that DIRC1A looks like a very, very good target. So I wonder whether you know, or anybody else know, but systematic studies to try to obtain something, obtain a molecule to knock out the kinase activity, and like say artificial intelligence study to build up the appropriate inhibitor. Do you know of any such a study?
In our case, we have been using ECC, as you know, is an inhibitor D1A. But it is also true that this kinase controls large numbers of cellular processes. And the big question that I observed that John de la Barre was also posing is that which degree of specificity we want to the inhibition DIR1A. because we need DIR1A, and we know also that even in trisomic models, if you have an excess of inhibition of DIR1A, things are not going well, and they're very bad. Then the problem is to find the equilibrium of which degree of DIR1A inhibition you want. I mean, ECCG was interesting because it was a weak inhibitor of DIR1A, and part of the activity of ECCG was, let's say, supported by other interactions with steroid receptors and others, okay? We don't know, and this is the big question, is what will happen with these extremely specific inhibitors of DIR1A if they are able to fully inhibit this kinase. The dose, of course, is going to be an issue, but this is something that we can, in pharmacology, we can solve, but we don't know that this is really a very good solution. The full inhibition of DIR1A.
You need the modulation, yeah.
Yeah, that's correct. Then cross your fingers. I hope that Manros are going to survive, and in the development of DIR1A inhibition. But let's say I'm skeptic a bit, because we already have been in animal models, the excess of inhibition or the excess of specificity in the inhibition is not always good. On the contrary, it's really bad for people, because you need it. I mean, you have 2 copies, and you need these 2 copies. It is true that people with Down syndrome, they have 3, but an excess in inhibition is going to leave you with maybe 1.5 copy. I mean, we cannot do mathematics like that, but it's just to have some kind of picture whatsoever.
If I may, Marcia, I see that there is an interesting question in the chat. Can you look?
Yeah, maybe this one I missed about.
By Murray.
Yeah, the need of giving very early treatment. So there was a question from Effie Murray about, the possibility to treat mothers during pregnancies as some of, the new approaches for early treatment.
When we were working with ECCG, I remember that in Paris they did some experiments showing that pregnant mice treated with ECCG, trisomic pregnant mice treated with ECCG, the offsprings were improving, and we tried to think about this in humans, but the performance of these studies in humans are very difficult. Not only for regulatory issues that, as you can imagine, are not easy, but also because the promise. I don't know. For example, with ECG, there were some concerns of maybe some cardiovascular activity that was could be an adverse effect for the mother. I mean, it's not. What I globally agree is that it's true that regulatory agencies, they ask us to demonstrate efficacy in adults, and then we are allowed to move to pediatric population, and even, let's say, even to infants, because in fact, probably this is the good situation. I mean, we are talking about a neurodevelopmental disorder. Then as soon as you start treating this disorder, much better should be the outcome. But this takes years, I mean, to be allowed to do these treatments in infants. And on the other hand, also, the demonstration of efficacy is very difficult, extremely difficult. I mean, as you can imagine, your level of communication with these infants or children is not easy at all. But in addition to that, there were I remember that there were some safety issues administering these kind of inhibitors during pregnancy, not when you have your offspring, but during pregnancy, there were some safety issues that maybe they are not applicable for all drugs. But I remember that in the particular case of the A1A inhibitors, there were some safety issues that in some way they say put the brake in the development of these studies.
Can I make a question? Hello? Is it
Giorgio,
possible to make a question? Yes, thanks. Hello? So
Marzia, sei senza microfono. Marzia, you're without microphone.
Okay.
Please,
Sorry,
Giorgio.
Giorgio. Thank you. Sorry.
Giorgio, you are without a microphone now.
Now we switched the microphones.
So now it's on, right? Thank
Okay.
you. So you mentioned that there are some issues and some experimental data about transcranial direct current stimulation, and you did not mention about transcranial magnetic stimulation, TMS. And I think that could be an interesting possibility, but since there is in Down syndrome a normal excitability, normal cortical excitability, but there are failure in synaptic plasticity. But I'm not sure why there is not literature about this. So do you think there are failure as well as?
That's not true. I mean, in the tested study, we used TMS but not for stimulating the brain, but to measuring brain activity. You know what I mean? Right. Well, the problem is that I would say this is the first time, and because we have an officer that loves us, that there are some fundings in the European Union supporting research in Down syndrome. And it is true that there is the program, for example, the GODS21 that is interested in the interplay between obesity and cognitive functioning, and there is this development of this molecule. I mean, and when you think why EU is supporting the clinical development of drugs, you say, why? This should be done by the company. Why we should support that? I mean, we're in a situation that now we have the possibility to have access to funding for Down syndrome that this was not available in the past. But who is going to fund your studies on TMS or TDCS or whatsoever? Nobody. Full stop. That's a problem. I mean, there is no money for Down syndrome. Down syndrome is in the border between the rare disease and the most prevalent diseases. And up to date, at least in Europe, Down syndrome is in the most prevalent disease, and our league is against Alzheimer's disease, cancer, cardiovascular disease. And as you can know, for people working on Down syndrome, we are just irrelevant. That's it.
Thank you. Thank you, Rafael. Sorry, but we are missing a lot of people just leaving, and we would like just to make conclusion for the session. The discussion is very interesting, and I hope it will give you any interaction or any comments in the future. But Renata and I, Lucio, of course, and the rest of the task force would like to thank you, all the audience. First of all, thank all the speakers for their wonderful presentation and for all the inputs we have discussed and shared this afternoon. And I just give Lucio the need of making some comments and conclusion before a meeting tomorrow morning.
Oh, that's it. I mean, thank you very, very much. Thank you to the speakers. Thank you all for being here, and we see tomorrow morning, 8:30. Okay. Thanks to all of you. Thanks.
Thank you so much.
Thank you.
Thank you very much.
Bye-bye. Bye-bye.
Thank you. Bye.
Bye-bye. Thanks.
Bye.
Bye.
Gianfranco, ci sei? Gianfranco, sei ancora lì?
E qui sono.
Filippo, caro.
Io vi saluto,
Grazie
è
a
stato
tutti.
bene.
Ci vediamo.
Grazie Marzia, grazie assai. Filippo, ti ho scritto un messaggio.
Sì, io avevo già mandato a lui
Ok,
le
ok.
nostre, ecco, alle
Un
15 l'ho
grazie
fatto.
davvero, ecco,
No, ma
grazie
perché
davvero
lui
speciale.
così,
e,
glielo faccio
diciamo,
arrivare di nuovo.
ha fatto anche
Grazie.
una conferenza estremamente interessante.
Va
Vi
bene,
prego di fargli
grazie.
avere
Lo
ulteriori
faccio subito,
apprezzamenti da
a
parte
domani.
nostra. Grazie.
Grazie a voi.
assai.
Ciao.
Gianfranco, tu ci sei? No.